keyboard to this computer. All right. Hi, everyone. Thank you so much for joining us today for the Insect Welfare Research Society's virtual seminar series. I'm delighted to be introducing Dr. Bob Elwood, who is going to be speaking to us today about crustacean pain. He's an emeritus professor in the School of Biological Sciences at Queen's University Belfast in the UK. Just a couple of notes before we get started. It is possible that Dr. Elwood's Wi-Fi might drop out briefly, in which case he'll return to us in just a moment. And if you have any questions as we're going through, you can feel free to look at the bottom of your screen. There's a little box that says Q&A, and you can drop those questions in the Q&A for me to ask them of Dr. Elwood when we get to the end of the presentation. And with that, I will hand it over to our speaker. Thank you so much.
Well, thank you for having me. So the question today is, do crustaceans feel pain? And why is this question important? It's important for two reasons. First, individual animals can be given most appalling treatments. They can be boiled alive, they can have their claws ripped off, they can be bound and transported for weeks totally immobilized. The other reason is there's actually huge numbers of these animals being used, so it's severity and number. But first of all, I'm going to give a little history of my work to ask, how did this question come about? What brought it to my attention? Well, I joined Queen's University Belfast back in 1975, seems just like yesterday. And as a lecturer in zoology and animal behavior, I could just follow whatever I wanted and the world was my oyster, and indeed I did study oysters at one stage. But this is a small array of the animals on which I've worked and published, and it's about half the species. In fact, in this picture, I couldn't get any more on. But there were two main groupings. First of all, there was rodents, and I did my PhD on maternal behavior and paternal behavior in rodents. And I pursued that when I got to Queen's, and for about 20 years, that was my major work. And then there was hermit crabs. I've fallen in love with hermit crabs when I was quite young. And I started pursuing a research project on hermit crab behavior. And that in fact took over really. So that by 1995, my main research really involved contests, how animals competed for resources. And I was asking, how do animals gather information about those resources and about the opponents, and how those changes altered the motivation and the decisions about fights. So that's where I was in the early part of this century, feeling quite satisfied with the way that my work was going and not thinking about pain one jot. So one night, around about 2004, I went down to my local pub, which I miss. I've moved from Northern Ireland. I now live in England. My local pub restaurant, and this is some of the sort of clientele you get in the small bar there. Everyone chats in there. And who should walk in but Rick Stein. Now, Rick Stein probably means nothing to you in the USA and maybe other parts of the world. But in the United Kingdom and Australia, Rick Stein is a superstar, a TV chef, has series after series. And he's mainly known for his seafood cooking. And so I chatted to him. We were both waiting for dinner one Friday evening. And I chatted to him about my interest in crustaceans and noted that he cooked them. And he simply asked, do they feel pain? Which I thought was a silly question, but I was polite and we chatted for a while, a bit longer. And I thought about it and sort of wondered if there was a way you could actually answer that question or get some sort of insights into it. So let's think about what pain is then. Most animals will respond. I've got a bar blocking my view. I've got rid of it. Most animals have nociceptors, and nociceptors respond to noxious potentially damaging stimuli. And these receptors initiate a reflex that removes the animal from the stimulus. So the animal avoids further damage. And we can see an example of that, or not clearly, but in this Norway lobster, or Dublin Bay prawn, sometimes called, if a noxious stimulus affects their front end, they flick the tail down, and the tail is flicked forwards like this, and the animal darts backwards away from the stimulus. And that can be shown to be a reflex reaction. And the animal thus escapes from the noxious stimuli. It stops the damage in the immediate sense. So in pain, in humans also have the reflex. If something hot touches your hand and you weren't expecting it, you move your hand very rapidly. And so we have reflexes. But the nociceptors in humans also link to the brain. And here it initiates an unpleasant experience that we call pain. Now, one thing you have to note about this is that the reflex occurs before the impulses get to the brain. The impulses to the brain are quite slow. Those conducting or organizing the reflex are rapid conducting. So the reflex occurs before pain. You don't move your hand because it hurts. You don't feel it at the time that you move your hand. So that's an interesting point, but it also poses a problem in that reflexes and pain are not the same. If you see an animal respond to a stimulus, it might be just by a reflex, and there's no implication of pain in when you see just that. It's a problem for investigating pain. Well, let's ask then, what is the function of pain? If the reflex is good at stopping the stimulus, then why have this unpleasant experience? And the only rational reason is that because it is unpleasant, humans at least will change future behavior to avoid that feeling. So pain enhances fitness by reducing future tissue damage and by helping recovery. These are the two ways it can enhance fitness. Well, with humans, studying pain is made somewhat easier in that we can ask them. We don't know necessarily whether they're telling the truth or they might be exaggerating, but at least we can get some indication from humans in general. But with animals, you can ask them all you like, but they won't answer. So we have a problem in studying pain in any animal. So my approach was to see if I could discriminate whether the behavior that these animals showed could be explained by a simple reflex response, or whether there was something to suggest that it was more than a reflex and thus possibly pain was being involved. So I looked at two aspects really. I looked at long-term changes in behavior that enhance fitness. And if I found those, that would support the idea of pain, wouldn't prove it, but it would support it. And also looking at short-term responses that clearly involve decision-making rather than a reflex. And then again, that would support the idea of pain. But even if I show both of those, it still doesn't prove pain, but it's suggestive of pain. And today I'm going to look at 5 criteria that might help discriminate between pain and nociceptive reflexes. And I'll go through these when I get to them. So prolonged directed rubbing. I mean, you all know that if you bang your finger or something, you tend to hold it, you direct attention to it, and you rub it and protect it. So this was the first experiment that I did following my chat with Rick Stein. We used these glass prawns, which are wonderful animals, extremely hardy animals, which is why I chose these to use them in the lab for the first experiment. And they have long antennae on which you can put various substances on and do things to the antennae. And again, that makes it a useful animal. So it was a two-stage experiment. The animal was taken out of the water, placed on damp tissue paper. One antenna was selected at random, and it was brushed with seawater or a local anesthetic. The animal was then put back into the water for 5 minutes and observed, and it was taken out again. And the same antenna was treated with water or sodium hydroxide or acetic acid or pinched with forceps. So if we look at the data, get straight to the data, following that second treatment, this is the grooming behavior. When these animals groom, they have tiny little pincers on their front legs, and they repeatedly pull the antenna through those pincers. They also showed a second activity, which I'm not going to show in the talk today, but it was almost exactly the same effects of rubbing the antenna against the side of the tank, a distinctive behavior, rubbing against the glass on the tank walls. So if the first treatment had been with water, and then the second treatment was with water, there was virtually no grooming or indeed rubbing. If the first treatment was water and the second treatment were these noxious chemicals, we see a dramatic increase in grooming, and we found the same for rubbing. And the data for pinching were not very clear at all. They didn't respond a lot. I think technically it was significant, statistically significant in the experiment, but not particularly interesting from the amount that occurred. If, however, we look at the behavior towards the alternative antenna, that is the antenna that had not been treated, we can see that grooming and rubbing was low towards that antenna. Now, that tells us that the noxious chemicals have not stimulated generalized grooming. It was directed towards the specific antenna that had been treated and with the noxious chemicals. If we look at the effect of pre-treatment with a local anesthetic, we see that here, this is the treated antenna having these treatments applied after the anesthetic. There is a significant reduction in grooming and also rubbing against the glass tank. So we see these two features: directed grooming at the specific point of the stimulus, and that is reduced by pre-treatment with a local anesthetic. In another experiment we did, but it's only been published quite recently, we sort of injected a rear walking leg, one of the walking legs at the rear, with water, 0.25, 0.5, or 1% acetic acid, rather than brushing it on an antenna. Now we're injecting it, and this the chemical caused these animals to hold the leg vertically, almost vertically. Quite an extreme behavior. You never see it in other contexts. They also rub the leg, that specific leg, with the adjacent legs on either side. So, and also with the higher levels of acetic acid, not the lower, the higher levels of acetic acid, we saw autotomy of the injected leg. And that is the leg is cast off. And I don't know for sure whether this autotomy can be attributed to be mediated by pain or suggested, but because it occurs at about the time that it's rubbing or holding the leg vertically, it's built in with that response, the behavioural response. And it is a sort of a behavioural response in that it triggers specific muscles that cause the leg to be cracked off at a specific joint. And there's no bleeding. It's a feature of many arthropods that they can do this. We see prolonged grooming, rubbing, and guarding. We see guarding in if the edible crabs are wounded, they guard the wound, and hermit crabs that have been had electrical stimulation on their abdomen, and if they get out of the shell, they will often turn and rub the abdomen, which we never see that with the hundreds and hundreds of hermit crabs that we've got out of shells by cracking the shell. They have never rubbed the abdomen, but they do if you put an electric shock on them. So these activities are all consistent with the idea of pain. They don't suggest a reflex at all. They are consistent, but they don't prove pain. I keep coming to that point, that difficulty of proof. So let's look at avoidance learning. And in this experiment, I used shore crabs. And shore crabs during daylight on the shore at low tide hide under rocks. They find dark crevices to hide away from avian predators. And the rock is important. This hiding point is important for their survival. So what we did was to put the crabs into a light environment, and it was brightly lit. But there were two shelters. These are our pretend rock crevices, so the crab went in, and it had a choice of crevices. We could deliver shocks to the hind legs of crabs, small electric shocks, by using insulated wire. And where we wrapped the wire around the leg joint, the insulation was taken off, so we could provide a reliable stimulus to these animals. When we put the animal in, every single crab that we experimented on, when you put it in, certainly for the first time, they will immediately run to one or other shelter. So it's a great reliable response, and they don't seem to have this response at random. There is a tendency to go one direction or the other. But what we did to take that into account was we randomly selected the crab to receive a shock or not on its first choice of shelter. If it remained in the shelter, it was shocked again 5 seconds later, and we repeated the shock every 5 seconds, and we gave 2 minutes per trial, 10 trials in total. At the end of the trial, the animal was taken out of the shelter, if it was still in there, taken out of the tank, and then returned into the middle of the tank, and it could take its choice of shelters again. Well, what we found was that what happened was on the second trial, the vast majority of crabs simply went back to their first choice of shelter. And being shot didn't affect that preference. But on the third trial, that is after 2 previous experiences, those crabs that were shot went to the alternative shelter. So they were showing rapid avoidance learning. Crabs also got out of the shelter if they were shot, only if they were shot. no crab in the non-shock shelter ever got out of the shelter. But here we see that even in the early trials, over 50% were getting out of the shelter. But this behavior increased. If they made the wrong choice, they quickly got out of the shelter. And that was a feature. They seemed to be doing two things. They were learning which was the right shelter, but if they got that wrong, they also seem to have learned, well, get out quickly, and that will stop the shock. So we see this swift avoidance learning. It's not predicted by a nociceptive reflex. And again, the data are consistent with the idea of pain. And there's since been a nice experiment on crayfish showing a very similar feature of learning very rapidly to avoid a preferred location. I'm going to look at motivational trade-offs. And to explain what I mean by this, let's simply say that reflexes don't require any higher level processing. It's not a behavioral decision, it's just a reflex. But behavioral decisions typically involve trade-offs between different motivational systems or requirements. So consider this non-arthropod example of zebra here, which every now and then, sort of every day or so, they come to a water hole or a river and where they drink. And they drink very reliably when they come to water. But there are things that inhibit them from drinking. And if there's sort of other stimuli there, so if there's a lion, a predator, drinking at the water hole, the zebra stand away and they're trading off. They're trading off their need for water and their need to avoid predators. And that shows that drinking in zebra clearly is not a reflex, it's a behavioral decision. These animals have a high motivation to drink, but an even higher motivation to avoid the predator. So if we see this, we know it's not a reflex behavior. So we did an experiment here with shore crabs again, and again we had an enclosure, but this time we just had one dark shelter. They could go into it or they could stay out of it. And we gave repeated shocks in the shelter. Sorry, we didn't give. We gave one shock when they went in the shelter, and or they had no shock. There were two levels of shock. There was 6 volts, 12 volts, or no shock at all. And the other variable in the experiment here was in the nature of the light. In half of the animals, they were put into arenas with very bright light, and in others, it was very, very dull light. So we had a variation here. And the measure here is the number of shelters that they entered in the last 5 trials of a 20 trial experiment. So the maximum here is 5, and that's what we see. These here animals here, virtually every animal went in on every occasion when there was a very bright light and there was no shock given. But a 6-volt shock substantially reduced the probability of going into the shelter, and a 12-volt shock reduced it again. But the other thing you see is that the light has an effect. The dull light, they are less likely to go into the shelter. But again, they're being influenced by shock, but they're being influenced by shock and by light levels. That is, they're trading off. They're making a decision based on memory of what's going to happen and the immediate light conditions. So they are trading off these two features of avoiding shock. This experiment, the animals have the choice of moving into a shelter that they might get a shock or not. They've learned that they will in some circumstances. but we can also do similar experiments where they have an immediate response to the shock. And that's what we did with hermit crabs. We are looking at crabs which we've wired up their shells. We drill holes in shells, wire them up, and so that we can deliver electric shocks to the abdomen. And we then offer one of these shells to a naked hermit crab. It gets in, and the crab looks like this. And where these wires are about where it places its naked abdomen. So we're not having to go through the carapace, which is calcified. We can apply electric shocks directly to the naked abdomen. When you do this, some crabs get out of the shell, and it looks as if it could be a reflex, because you just see them scrabbling out of the shell and sometimes running away from the locality of the shell. But what we did in one experiment was to have the animals in a small enclosure, and some of the animals had the odor of a predator present, and another group had an odor of a non-predator, and others just had seawater. And this is the percentage of each experimental group that got out of the shell to standard shocking. And you can see that 41%, only 41% got out when there was a predator odor, but 95% got out when it was just seawater, and it was about the same also for a non-predator odor. So these two are both different. Sorry, this one is different from both of these two in terms of the probability of getting out. They are less likely to get out if the odor of a predator is there. So this is just like the zebra example, if you like. They are moderating their behavior towards the electric shock in order to avoid potential predators. So we see there is this trade-off. So getting out of the shell can't be a reflex. It's a behavioral decision that's taking the shock and the predator into account. We see the same thing with choice of shells. The shells that the crabs use in Northern Ireland are the small crabs, at least, are these Littorina shells, and they really like these Littorina shells, and they really don't like these gibular shells. We wired up shells of both species and delivered shocks to these crabs. And we found we didn't want them to get out for this particular experiment, but a few did get out, but significantly more got out from the poor quality gibular shells. So again, they're trading off between avoiding the shock and keeping a quality shell. I'm going to move on to the long-term behavior change, because this is the same experiment, in fact, as that example with the two species wired up. We wired up these two species. We delivered shocks, in fact, to some crabs, but not others. And if they didn't exit, they went into the next part of the experiment. So if they remained in their shell, they were subsequently offered a new shell, an empty shell of the same species, and was asked, has your motivation to change shells been influenced by having a shock in the shell? And we found that the behavior changed. They were more likely to approach the new shell and more likely to move into the new shell. This is the latency to contact the shell. If they weren't shocked here, they showed a much longer latency to if they had received shocks. They seemed keener on getting this new shell. And hermit crabs invariably probe into an empty shell with their chilipeds, their claws. And what we found was that those that were shocked use fewer chiliped probes than those that were not shot. So they show a much higher motivation to get a new shell if they've been shot in their existing shell. What we found in the first experiment we did, we offered this new shell just 20 seconds after the shock was given. But we did other experiments with a much longer gap, and they were still showing this behavioural difference, this higher motivation to obtain a new shell, 24 hours after receiving a shock. So this is a long-term change in behaviour. It cannot possibly be described as a reflex. It's something that's happening 24 hours after the stimulus has stopped. And it's showing that they have devalued their shell. It's as if they all have rubbish shells that they behave like crabs in very, very poor quality shells. So we see that dramatic change. Anxiety. I'm going to move on quite quickly and skip that experiment that was done, a beautiful experiment done by some French workers. Just be egocentric and tell you about one that we did. In the experiment where we had crabs, shore crabs, with a single shelter that were not getting shock, a 6-volt shock or a 12-volt shock, one thing that we noted was that the latency to enter the shelter, if they did enter, these are only crabs that entered the shelter, those that were receiving shocks showed a longer latency to get into the shelter. And you might think that maybe they couldn't walk because they were slowed down, physically slowed down because they were shot. But what we found was when we looked at an alternative behavior, that is, they seemed to back up and push against the walls on the corner of the tank, trying to find another way of hiding or escaping the situation, and only then going into the shelter. So it's these animals that were being shot are performing this alternative activity. And it seems it's sort of an anxiety. They are trying to avoid going into the shelter, but they really need the shelter to survive because it's extremely dangerous for these animals to be in light conditions. Now, I alluded earlier on to the benefits of pain, and one thing that one experiment on crustaceans by some French workers showed that exposure to shock increased their shelter use. Animals that were shot now sought shelter more effectively, and if there was a fish predator, animals that had been shot showed a better survival because they were hiding in sort of crevices and avoiding the predatory attempts of the fish. So there is some fitness gains from this sort of anxiety, if you like, this change in behavior following electric shock. So these data cannot be explained by nociceptive reflexes. There are long-term behavioral changes after exposure to noxious stimuli, and these are consistent with the idea of pain. There's an awareness of the site of injury. Hermit crabs and shore crabs that leave their shelters are giving up valuable resources to avoid the stimulus. There are indications of anxiety. There's an increased attention to danger. And while all these are consistent with the idea of pain, that doesn't in fact prove pain. Quite annoyingly, but there we are. However, the accumulation of evidence so far suggests that we can't dismiss the idea of pain in these animals. the old idea that everything they did was simply by reflex, I think, has been rejected. We should give the animals the benefit of the doubt. It's known as the precautionary principle. They should be treated with care in experiments and should be slaughtered in a humane manner. Now, it was 17 years ago that we published our first paper on pain in decapods, and I was staggered by the responses of the media to that paper and subsequent papers. I really hadn't prepared myself for the attention that would be given to this. I was just a scientist trying to answer a scientific question, and I assumed it would be, discussed at conferences and maybe in scientific articles. I hadn't been ready for worldwide TV, radio, and newspaper coverage, which took me by surprise. Other things started to happen. A group called Crustacean Compassion was started up in 2017 and started campaigning for better treatment of crustaceans. The Humane Slaughter Association changed their views and started to support and promote suitable slaughter methods for crustaceans. The British Veterinary Association drew up a position paper saying that these animals should be treated humanely. The Royal Society for Protection of Cruelty for Animals, which is very big in the UK, drew up a position paper for protection of crustaceans. PETA is the only group that were campaigning against the use of crustaceans in cooking and human food, but they increased their objection to that. And there are a variety of other groups. Switzerland banned the boiling of live lobsters in 2018, 2019, I think. And there was a bill being brought in the UK Parliament declaring that vertebrates were sentient and so organising their better treatment. But an amendment was brought in to include cephalopod mollusks and decapod crustaceans as being sentient animals. And the government commissioned a report, a very extensive report on these two groups and the evidence for sentience. And lo and behold, the amendments passed. And so the UK government has now declared that these animals are sentient. There's been much advice following on from that on humane killing. There's a device called Crustastun was developed. It was developed actually before my work was published. I have to be clear on that. But it has been promoted extensively since then. In providing a way that animals can be killed effectively in microseconds. Once the power is turned on, the nervous system ceases to function within microseconds. There is advice on people that don't have access to crustaceans but are using these animals in the food chain in restaurants is immediate destruction of the central nervous system or very rapid destruction. It's a second choice to electrocution. And there's advice not to boil live lobsters or other crustaceans. And now there's been changes in the attitude of UK retailers. They have stopped selling live lobsters and live crabs. They are now demanding that their suppliers use more humane methods. At least some of the retailers, I have to say, not every retailer is doing this, but some of the more reputable ones are doing this. And now there's moves in the actual fishing industry, the industry where you might expect to see the most resistance, where it might have some economic impact. It shouldn't be disastrous, but they are being asked to improve their methodology so as to improve the welfare of these animals, again, particularly about commercial killing in large numbers. And this is where the numbers really come in. It's the commercial fisheries that are important. So we've had a whole range of responses, and things are changing quite dramatically. So I just like to finish this talk now by thanking you for the invitation, and to the students, and to Rick Stein for asking the question in the first place. So thank you. I'm very happy to answer questions.
So thank you so much for your talk. That was fascinating. We do have some questions in the chat, and I'll just encourage everyone in the audience, if you have questions, you can go to the bottom of your screen. There's a little Q&A button, and you can type your questions in there, and then I can ask as many as we have time for to Dr. Elwood. I am going to quickly just pause the recording or stop the recording for this part, and then we'll do our Q&A.